Today we're going to do the chapter on inherited change. This is the syllabus code 9700 and this is the A2 syllabus. We're going to talk about number 1, the Lac Operon. We're going to talk about Gibberellins, and we are possibly, if we can accommodate it, the transcription factors. First of all, we need to know that what are we talking about? We're going to be talking about a bacterium has this bacterial chromosome. Now a portion of it is called an operon. A portion of it is called an operon. What is an operon? An operon consists of 3 genes. Now here what we have, gene 1, gene 2 and gene 3, and these work together. So if transcription takes place, then these 3 genes are going to be resulting in the production of protein 1, protein 2 and protein 3. So we have transcription and then translation. So but first mRNA must be made. So the mRNA is made. So this is the DNA. This is on this bacterial chromosome. This is a portion of it here. And before that, what do we have is we have a promoter. So there is an area of the DNA where of course we have A, T, G, C, nothing else, is the same nucleotides. But that area, which is a specific area, is called the promoter. And then this area is called an operator. So looking at another diagram of this, the Lac Operon, what do we have? We have Lac ZYA, Z, Y and A, and these are called structural genes. Just another diagram to explain the same thing which I did earlier. And then you see this is the promoter for the structural gene. This area, the pink one. And this purple is the operator. So the promoter plus the operator plus Z, Y and A, this is called the Lac Operon. But before that, I want you to also understand what is, this is the promoter for a regulatory gene, and this is the regulatory gene which codes for a repressor protein. And a repressor protein, the word you can understand, means something which is going to be repressing something. We're going to be talking of 2 things. Number 1, inducible, which can be induced, and repressible. So please understand these 2 words before we continue with the details of this Lac Operon. Inducible, which can be made to be produced, some sort of a protein or an enzyme. Repressible means something which is being produced but then can be stopped. So inducible, something which can be induced to be made a protein or an enzyme, which when a substrate is present, and repressible means it's happening regularly, normally, and we need to stop it. Another diagram explaining this phenomena. We have here the Lac Operon. Now promoter, operator and Lac Z, Y and A. Now, so the promoter is where the RNA polymerase sits down. So here the RNA polymerase is going to bind. The Lac Operon proper is Lac ZYA. These genes have to be on the same strand because you're making one RNA, and the coding and the template strands have to be consistent when transcribing certain genes. They also have to be the same because they're made from the same RNA. So there's going to be an enzyme which is going to be made here, then there's going to be an enzyme which is going to be made here, and then there's going to be an enzyme which is going to be made here. Now just a few key points. Bacterial genes are found in operons. Genes in an operon are transcribed as a group and have a single promoter. Each operon contains a regulatory DNA sequence, just like you have a switch for a fan and then you have a regulator for the fan. So the switch is going to just switch on the fan, but your regulator is either going to have it going at a faster speed or a slower speed. So each operon contains regulatory DNA sequences which act as binding sites for regulatory proteins that promote or inhibit transcription. You know what was transcription? Transcription was DNA to mRNA. This is the first stage of protein synthesis. Number 3, regulatory proteins often bind to small molecules which can make the protein active or inactive by changing its ability to bind DNA. Next, some operons are inducible, meaning that they can be turned on by the presence of a particular small molecule. So inducible operons and others are repressible operons, meaning that they are on by default. They are on by default, but can be turned off by a small molecule. So please understand these key points and then proceed on to the next part that we are going to be studying in detail. Now the next step that we have to understand is that bacteria are unicellular. They have a cell wall, they have a cell membrane, and they have a circular DNA inside it. And this is the area where this Lac Operon is present. And the bacteria cannot afford to waste its amino acids. Waste its amino acids means make proteins which are not needed by the bacteria. So if the bacteria is in a glucose solution, it doesn't need these 3 enzymes, LAC-Z, LAC-Y and LAC-A. So it will not make it. But how does the bacteria know it's in a glucose solution? Now this is what is happening. Now we've got to understand this. There's an active repressor protein. And this repressor protein binds to the operator. There is a repressor protein which binds to the operator. So LAC-Z, LAC-Y and LAC-A are not going to be made. This is the situation when lactose is absent and the bacteria is in a solution of glucose. So now you can see here the RNA polymerase is sort of being stopped. The RNA polymerase cannot do its work. It cannot make the mRNA for LAC-Z, LAC-Y and LAC-A. So the protein, which is the repressor protein, is binding to the site which is the operator site and is preventing the transcription of the mRNA for these 3 enzymes. So no mRNA made, no enzyme is made. But look what happens when we take this bacteria and we put it in a solution of lactose. We take this bacteria and put it in a solution of lactose. And what happens? The lactose binds to the repressor. You can see this happening here. The lactose binds to the repressor. Now this is an inactive repressor. So it has come out of this place and it has moved out. We've moved it out of it. Look at it here and then look here. There is no blockage now happening. So when there is no blockage now, what can happen now is that the RNA polymerase, this is the RNA polymerase, now this RNA polymerase can bind and you can see now mRNA is being made. And what are these three enzymes? We've got to know the names. LAC-Z is called beta-galactosidase. Beta-galactosidase. LAC-Y is called permease. LAC-Y is called permease. Lac A is called transacetylase. Lac A is called transacetylase. So we have to know these three names, and these three enzymes are needed so as the bacteria can break down the lactose into its two constituent parts, glucose and galactose, and then this bacteria can use this or burn it as a fuel and use it as a respiratory substrate. The important subject which the topic we need to understand is, number one, some structures which are called, like these three structures are called structural genes. But then we also have a regulatory gene, and the regulatory gene is LAC-I, which actually results in mRNA, and it results in this protein being made, which is called an active repressor protein. And this active repressor protein goes and binds to the operator and prevents RNA polymerase from binding and forming the mRNA. So we have to understand is that the regulatory gene. So we have to understand the two words regulatory gene. So just like I told you before, there's a regulator for the fan, regulatory gene. So these regulatory genes actually regulate and ensure that transcription occurs only when those proteins are needed by the bacteria. So the bacteria does not waste its amino acids. So the difference is regulatory gene and structural genes. Structural genes are usually the ones which are used for the bacteria uses it for making certain enzymes or proteins which are beneficial for its normal functioning. Now we come to the second topic, which is that of gibberellins. So gibberellin is also called gibberellic acid. Now what we need to understand is that what happens in a barley seed when we want to promote germination. What happens? Now first of all, we've got to go back a little to O-level biology. What are the factors necessary for germination? And I usually give you a very simple mnemonic to remember. It is TWO. T is for temperature. Every seed would need an adequate temperature. And please remember, we need water. Do not say moisture. Moisture is incorrect. Moisture is incorrect. Sometimes you all write moisture and that's incorrect. And we need oxygen. So oxygen is needed. Please do not say air. Air would be wrong because air has a lot of other stuff in it. So factors necessary for germination are temperature, water and oxygen. Now going back to the barley seed, first of all we say water uptake initiates germination. Water uptake initiates germination. Number 2, embryo synthesizes gibberellin in response to water uptake. Number 2, embryo synthesizes gibberellin in response to water uptake. Number 3, the gibberellin diffuses to the alluron layer. Now you can see the gibberellin here is moving to the outer layer, which is called the alluron layer. Now this alluron layer synthesizes amylase. Now how does it know that when I have to make amylase and when I do have not to make amylase? You have barley seeds lying in a bottle in your kitchen. Well, it's not going to make gibberellin, it's not going to make any amylase, but suddenly when you soak the seed in water, we find amylase starts to be made. So that means transcription has started to occur. Because please remember, amylase is an enzyme, so it's a protein. And we said protein synthesis is when DNA will result in the gene being copied and mRNA being made. So transcription is DNA to mRNA. So number 1, I said water uptake initiates germination. Number 2, I said embryo synthesizes gibberellin. This gibberellin number 3 diffuses to the alluron layer. Number 4, alluron layer synthesizes amylase in response to gibberellin. Of course, what happens to the amylase? The amylase is going to come into the endosperm, convert the starch to maltose, maltose to glucose, glucose is going to be respired, and the radicle is going to grow down, and the plumule is going to grow up, and the plant is going to start to, the little seedling is going to start to be growing. But what we need to understand is, how does gibberellin know, and how does gibberellin result in the formation of amylase? So we've got to now correlate this and understand how does this happen. Now, as you can see here, you can see how this is actually brought about. How does the gibberellin promote amylase production? Now, the first point we've got to understand is, let's look at this diagram very carefully. You have a gibberellin receptor and you have a gibberellin molecule. So the gibberellin which is produced by the embryo moves to the alluron layer. Now there must be receptors on the cells of the alluron layer where this gibberellin is going to go and fit. Now when this fits, number 2, what happens? Now the gibberellin bonds with its receptor, which is the green one, and an enzyme. Now yellow is the enzyme. Now this results in the destruction of this protein which is called DELA protein. Looks like Della is a girl. So DELA protein is destroyed, it breaks up. But what has been released is the PIF. And what is PIF? PIF is phytochrome interacting protein. And now this PIF can now bind with the promoter area, and transcription can be initiated. Transcription of what? Transcription of amylase. Again, once again, I'll just repeat myself. Gibberellin binds to its receptor. So PIF cannot bind to the gene promoter while it is bound to the DELA protein. So this PIF is actually bound to the DELA, and this cannot work. So what has to happen is that when the gibberellin binds to the receptor binds to an enzyme. The enzyme results in the destruction of the DELA. Now this PIF is free. It can bind to the DNA, and it is going to cause the transcription of amylase. So naturally, if transcription occurs, then translation occurs and amylase is going to be made. So the amylase is going to result in the starch in the endosperm to be converted to maltose, maltose to glucose, and the glucose will be respired and energy released. The topic, which is transcription factors in eukaryotes. You see, eukaryotes are, most of them, not all of them, are multicellular. So what we need to understand is that the proteins which are needed, number 1, are needed for the normal functioning of each eukaryote. And these proteins are mostly, we call them enzymes. Some hormones are proteins. So I won't say all hormones. Some hormones are proteins. Then we have structural proteins. Like you need a channel protein for chloride somewhere. You need a channel protein for calcium somewhere. And the important thing is, in the development of any organism, like in the development of a fetus, whether that fetus is of a human or is that of an animal, there has to be, the sequence has to be correct. How does this fetus develop? What are the organs which have to develop first, and then the second and third, and so on and so forth? So the sequence of development is controlled by all these transcription factors. Now if we look at the, this is a page from the book which has been, your A-level book which has been prescribed by Cambridge. effect of transcription factors include the following. Number one, general transcription factors are necessary for transcription to occur. They form part of the protein complex that binds to the promoter region of the gene concerned. You know, the promoter is the area just before the actual gene where the RNA polymerase binds. Number two, other factors activate appropriate genes in sequence, allowing the correct pattern of development of the body regions. Number three, a transcription factor is responsible for determination of sex in mammals. I mean, as you know, if it's an XY chromosome in the zygote, then the male reproductive organs will develop, the testes. While if it's XX, then the female reproductive organs have to be developed, which is the ovaries. Next, transcription factor allows response to environmental stimuli, like switching on the correct genes to respond to high environmental temperatures. Then, of course, we have another point, which is transcription include the products of proto-oncogenes and tumor suppressor genes, so that we can regulate cell cycle, growth, and programmed cell death, like red blood cells must die after a certain period of time. And then the last point is that hormones have their effects through the transcription factors. So these are the main points about transcription factors, and sometimes you get a very complicated question on this, which really checks if you've understood this very well or not. The next topic that I want to talk about is interactions between loci. Now, in some situations we have a very clear-cut dominant and recessive, and that's it. But sometimes some genes interact with each other. Now here what we have is in chickens, the inheritance of feather color in chickens, there is an interaction between the two gene loci, II and CC. Now individuals which carry the capital I have white feathers. Even if they carry the dominant allele big C, though C is for colored feathers, but if I and capital C are together, then they'll still be white. They won't be colored. There won't be a mixture of white and colored. Even if they have the big I, now this will be given to you in the question, that what is the interaction. So big I and big C both will result in white feathers of the chickens. And also if they are homozygous recessive, meaning both small i's and both small c's, even then they are white. Now this is something which is not, you're not supposed to know it, but you'll be told about this in the question. In some way you will be told this. So what do we have now? When we have the capital I, so homozygous dominant for both and homozygous recessive for both, when you have it, all are heterozygous for both. All of these are white. All the big I, small I, this is the one, big C, small c, they are all white. Now this is heterozygous for both characteristics. Now when we cross it with a heterozygous for both characteristics, if you remember from a previous, when we cross two heterozygous for both characteristics, we get a ratio of 9:3:3:1. But here we don't get that. Why do we not get that? Now that is what we've got to understand, is why are we not getting a 9:3:3:1 ratio? Because here we have interactions between loci. Now in this situation, if you can see, this is white here again, then this is white here again. Why? Because it's got another big C, then white again, then white again, then white, then white, then white. So 8 white, 9 white, 10, 11, 12 and 13. So there are 13 white now. And there are only 3 colored. You see, the ones which are colored are these ones. So the normal 9331 ratio is not present here. Why? Because this is an interaction between loci. So the 9331 ratio is not here, it is 13:3 ratio. Now it could be very different, it's not always going to be 13:3. Please remember that, it depends upon which interactions are we talking about. So interactions between loci is going to be, information is going to be given to you in the question. Like here it was told to you in the question that if the big I and the big C are present, then it's white. And if it is homozygous recessive, even then it's white. So this information has to be then put into the question, and you've got to understand how you're going to put it into the question and answer the question.